Hey, everybody. Good morning.
Good morning.
So nice and warm and cozy. I am not sad at all to be sitting on my couch, I have to say.
Professor, I have a question.
Yeah, far away.
The homework one, is that due on Saturday?
It's a good question. Let me check. Open up
Okay.
the schedule and see.
Thank you so much.
That sounds familiar, but let me check. Where are we at? 4th. Yep, that's right. Homework 1 is due on Saturday the 4th, midnight.
Okay, thank you so much.
No worries. I'm gonna turn my sound up a bit. Sorry, I still got bits of my breakfast in my teeth. I've only just finished, funny enough, with my bowl here. Unfortunately, I stayed in bed a little bit too long because it was cozy. Anyway, all right. I gather my thoughts. This is going to be a bit of an adventure because I'm going to have to do a whole bunch of Mendelian genetics on my tiny whiteboard. So if you have any issues at all, which you probably will, actually seeing what I'm doing, just let me know and I'll move the computer closer. Okay, where are we at? All right, so get my thoughts. What are we doing? So today we need to finish up the overview of Mendelian crosses, right? Because they are both monohybrid and dihybrid crosses are the same. We've already done the assumption, so we don't need to cover that. We finished that on Thursday. And then we'll get through a monohybrid cross at the very least. I can't imagine that taking us too long. And then we'll get started on a dihybrid cross. So whether we'll finish all of that today, I don't know, but we'll see. All right, let's see. It's been ages since I've taught my couch. This is kind of a blast from the past. Let's see if that's too far away. I've got one of those kind of like couch dining table things that my laptop is on. Out of the way. See how we do with reflections. If you have issues with the reflections off the whiteboard, let me know and I'll rejiggle things a little bit. There you go. That'll do. All right. So get my phone out too. To do today. Actually, I just spotted my youngest daughter. Nothing. I'll wait till she comes past, then I'll no, she's shaking her head. Nothing bad. I'm just going to introduce you to my class. That's all. Sure. All right. Temporary distraction. Blame my daughter. It's her fault. Let's see. Ta-da!
Daughter and dog.
There you go. Cricket is not amused. Evie is amused. Ta-da! There you go. All right. Hello, Cricket. You can't stay there though, love, okay? No. I've just fed them, so they should hopefully be somewhat calm. Anyway. All right. Like that, Victoria? They didn't get treats. They got their breakfast. That's more than enough. They're good. You know what? I'm going to close the blinds from outside because that's the other reflection from the window. Oh, yeah, that's good. Hockey schmocky. So, unfortunately, this is like a totally non-permanent solution, so it does move around a little bit. There we go. What was I saying? Cross overview, which is essentially 3 generations and their phenotypes. Can you actually read that? Yeah, more or less. I'll try and be better at writing. It's just hard because my arm is only ever so long. So, monohybrid cross, and then dihybrid cross. If we have time. If we don't time to get through all of that, that's fine. Also, as I said in the email, this winter storm thing is scheduled until to last until Thursday. So essentially my plan is to get up when the dogs wake me up, have a look out the window. If it looks bad, we'll be teaching online. We'll be learning online. I'll be teaching, you'll be learning. And I'll let you know in enough time so that even those of you with the longest commutes to class will get it before they leave. So keep your eyes open for that. I'm working on the assumption that we'll be at home until Thursday at least. But again, I don't know until I wake up in the morning. All right. There's the first one. Okay. So, crossover. All right, so I can't emphasize this enough. But, sorry. Every Mendelian cross happens exactly the same way. So things may differ, such as the trait that you're looking at in the cross, that's involved in the cross, the phenotypes that you're looking at, the particular alleles, but always they will be the same layout. So mono means one, obviously. One gene, trait in this case, 2 alleles. Right? One dominant, one recessive. Dihybrid, 2 genes, traits, each with 2 alleles. That's always the case, right? And you'll always have 3 generations. Parents. I can have enough space down there. Yep. Offspring 1. And then these, it kind of goes as almost like a flow chart, right? Offspring 2. That's the general idea. I'm going to go into the details of this particular part in greater depth in a second. So this is always the same. If it doesn't happen like this, it's not a Mendelian cross, or it's not a monohybrid or a dihybrid cross. And again, all these pictures will be going up to Blackboard, But if you do want me to slow down, pardon me, because I can't see you writing, right? So I don't have that sort of feedback anymore. Either unmute yourself and just shout out, "Whoa, soldier," something like that, or put it in the chat because I've got that up as well. Okay. Oh, see if I can show you cricket. Just, I don't know if you could see it just past that ornament. She's got a head on the other side. She's wondering why she can't come over and hang out with me, because that one is my spirit animal, even though other in the family have claimed it. So we always have a P0 parental. She wants to learn to. You want to learn genetics, cricket? I think she's hoping for more food. That's probably what she wants. So this is the parental generation. We have the F1, and these are the terms that we'll use, right? This is the first filial generation, right? So these are essentially brothers and sisters, offspring of the parents. These then mate with each other, which is fine if you're peas, and it's not too bad if you're cats, I guess. And that produces the F2, which is the second filial generation. Right, so it's always this layout, and this is so rigid. In fact, there are very few things in biology that are as rigid as a Mendelian cross. Can't actually think of any off the top of my head. But this is so rigid that I can give you any bit of information from any part of this, and you'll be able to work out the rest of it. In fact, that's quite frequently an exam question, right? So I could give you the phenotype of the F1s, and you can tell me the phenotypes and genotypes of the parents and of the F2 generation, right? Without even thinking about it. That's where you need to be at with this. All right? Because I will be quizzing you mercilessly on Mendelian genetics in the first exam, without a shadow of doubt. There's going to, because it's a big chunk of what we're talking about, it's going to be a big chunk of what I'm going to quiz you on. All right, so each generation has its own particular set of genotypes, genotype, and phenotypes, right? So the parental generation are what's called, I don't know what happened there. Hopefully you can read that. True breeding, right? Which is essentially the opposite of all of my dogs, because they're complete and total mutts, San Antonio specials. True breeding basically means that if you have, oh, what did I use in the other class? Miniature chihuahuas, right? Which I think exist. Not sure, but I've seen some really, really small chihuahuas. So if you have two true breeding miniature chihuahuas, I don't know, they'll be about like that big, I guess, then you'll only ever get miniature chihuahuas, right? Even if you take their kids, breed them to each other, and their kids, breed them to each other, and so on and so forth. For however many generations you want, you'll only ever get miniature chihuahuas, right? You won't get like a Labrador or something pop out. That's what true breeding means. And essentially what true breeding means in terms of genetics is that they are homozygous. So they have two of the same alleles for the gene that we're working with. Genes in case of dihybrid, right? So there's no other allele in there that can kind of pop out, right? That was previously masked and then can be seen again later on. It is only ever the same alleles. Okay, so homozygous, one's going to be homozygous dominant, right? So it's going to have 2 dominant alleles. And that's going to be crossed with a homozygous, recessive. I'm just using shorthand for space reasons. So one of them is going to be homozygous dominant, one of them is going to be homozygous recessive. I don't actually need to tell you which is which, right? And I'll get to that in a little bit. Now, that's going to produce offspring, which are all, so this is the F1 generation. They're all going to be heterozygous. Because that's the only genotype you can get when they get a dominant allele from one parent and a recessive allele from the other one. Does that make sense? All right, so here's a question for the class. What phenotype is the F1 generation going to express?
All dominant.
Going to be all dominant. Why is that, Mike?
Well, because the dominant overpowers the recessive, and if you have 2 homozygous, one dominant, one recessive breeding, the dominant phenotype is going to express that because it's dominant.
Excellent, exactly. So the heterozygous offspring, the F1s, thank you, Michael. They, I don't know, let's use letters A, right? Like so. They're going to have a single dominant allele, and because this is Mendelian inheritance, that allele is going to be dominant over the recessive allele, and so you're going to express the dominant phenotype. All right, so someone else, because I know you're all there, I think. Which parent is going to have the same phenotype as the F1s? Or another way of asking the question, which 2 of the 3 here, these individuals, are going to have the same phenotype?
The dominant parent and all of the offspring.
Exactly. Thank you, Matthew. So these, I think purple is the only one I've got left that works. So the dominant parent and the offspring, the F1s, have the same phenotype. So I can tell you what the phenotype of the F1s are, and you'll be able to tell me which of the parents is dominant and what their genotypes are. Right? Because it always happens exactly this way, no other way. All right. And so these then, does that make sense to everybody? It's a really, really important point. Right? It's kind of one of the keys to figuring out mono and dihybrid crosses. Yeah. Okay. So these individuals, the F1s, dominant heterozygotes, they're going to be crossed together, and that gives you a very specific. Oh, okay. Sorry, Haley. Just trying to think of a way of doing this without erasing any of this stuff. Let's see if I can do a drawing. You're going to regret this. whiteboard. Mm, mm, mm. Ooh. Yeah, no worries, Haley. It's totally okay. I'm just bloody hell, I don't know what that is. Like this is going to be like finger painting, right? So this might be a little bit torturous to watch. Hopefully I'll do a little bit better. All right, so and this will also make more sense too when we go through the natural example. Joshua, yes, but that's only for a dihybrid cross. Ah, yeah, there you go. Good stuff. I don't know if you can actually see me. I'm like gesturing here, or you can just see the whiteboard. Okay, so and all right, this was supposed to be a cat. It's now turned into a bunny. Oh, yeah, that's pretty good actually. There you go. That is Evie, you should see my finger painting for class. All right, so draw some eyes. That's supposed to be a rabbit. Very good one. All right, so anyway, this is the F1. There you go. My daughter says it's horrifying. It's supposed to be a rabbit. My finger said, "What do you mean? It's got a buffy tail and everything." Like,
if I looked at that and you didn't tell me that was a rabbit,
okay, well, okay, we've got an alien then. It's an alien F1. Right, so this is black. Oh, thanks, Mike. Michael, sorry. Drilled triangle nose. Is that an upside down triangle or a downside triangle? Like that? I don't think that makes it any prettier, Joshua, to be honest. But anyway, so this is a black rabbit. I suppose they exist. Right, I'm not entirely sure. There you go. Which was supposed to be a cat. All right, so genotype of this F1 rabbit is what? This is particularly directed to Haley, not because I'm picking on you, just because I want you to kind of work through this with us. Ah, Haley, that's the phenotype. What's the genotype? Pick any letter you like. So it's F1. Dominant phenotype. If we use, let's use B for bunny. What's the genotype? So remember, its parents are homozygous dominant and homozygous recessive. Yeah, thank you, Trinity. It's going to be heterozygous. That's right. It's okay if you don't know, Haley. That's totally fine. That's why we're here. Heterozygous. Sometimes it takes a, slightly different way of thinking about it or looking at it to kind of click or make sense. All right. So if this is, let me type this out. Oops, sorry. Eat off of that. So this is dominant heterozygous. All right. Question is, which parent is the F1 offspring it's like in terms of phenotype? So don't look at the genotype, right? Because the genotype is not what you see. Look at the phenotype. So remember, we've got two parents. I don't even know if you can see my fingers. One is homozygous dominant, the other is homozygous recessive. Which bunny, I guess, in the parental generation is the F1 that I've drawn here going to look like? What do you think? Think about it. We've only got two different phenotypes, Haley. You can actually get a 50% chance of guessing it right. The other one, love. So the homozygous recessive one, yeah, there you go. That was fast. Yes, exactly. The homozygous dominant one, because they're both going to express the dominant phenotype, right? So I'll type that out too. It doesn't hurt. Both the homozygous dominant parent and that in capitals, the heterozygous F1 offspring will express the dominant phenotype. Right, so you only need to know which phenotype the F1s express, and that tells you which is the dominant phenotype. Right, so that's essentially the kind of the point behind this. Did that help, Haley? Super cool, awesome. Thank you for asking the question. And oh, I don't know if you actually get to see that later. Hopefully that is lost to history, right? I'm not sure that any part of our culture will be improved by preserving that picture. Okay, but also, Haley, this will make sense when we work through it in detail too, right? So this again is the overview. We're not done by any stretch. We're going to go into a detailed look at a monohybrid cross in a minute, and that will also help too, right? So that's going to be another way of looking at the same information. All right, so then I think Joshua brought this up, right? So we get a very specific ratio depending on whether it's a monohybrid or a dihybrid cross. If it's a monohybrid, it's 3:1, dominant to recessive. And if it's a dihybrid, as Joshua said, it's a 9:3, 3:1. I'm not going to write out what these different numbers mean yet, because it's kind of complicated to write here without going across. So I'll leave that as it is as a placeholder for later. But either way, whichever cross you're doing, you would expect, you're not necessarily going to get exactly that, but you would expect a 3:1 ratio if it's a monohybrid cross, or a 9:3 to 3:1 ratio if it's a dihybrid cross, right? Okay, so let's actually get into a cross and we'll work through the different steps, and that should also help a bunch as well. Okay. Yeah. Okay, monohybrid. Oops. Oh, I have to spell this right. Okay, so already said, but it doesn't hurt to repeat it. One gene, two alleles, and because of the way the cross works, two phenotypes. All right. So what do I want to do? All right, let's put it to poll. Student participation. Do you want to have the trait of interest be fur color or fur length? So you got a choice. Color from Joshua. Anybody else want to chime in? Color. Wow, that's pretty convincing. Oh man, I actually quite like doing fur length. I'll do that one for the dihybrid cross. All right. Color it is by unanimous vote. Let me just roll back up. So Joshua, question about haplosufficiency. That applies to the, well, it's kind of a property of the allele itself, but also it refers to the heterozygote. So essentially, one is enough, right? Or half is sufficient, essentially, is what haplosufficient is really talking about. So half of the number of alleles working is enough to get function. That's what haplosufficient is all about. And the reason why I introduced that term now is because later on we'll be talking about non-Mendelian inheritance, and that includes haploinsufficient, which is a different kettle of fish. But we're not going to worry about that right now. So fur color it is. Bizarrely, because this is the only color pen that I have that works left, because I keep thinking I'm never going to be teaching on Zoom again, so I don't bring any home. We have purple cats and black cats. Okay, so here's another way of testing your metal. That's metal, M-E-T-T-L-E, obviously, not how much metal you have in you. All right. Actually, you know what? I'm not going to do it backwards like that because I don't want to confuse people. Let's do it conventional. So P0, we have, I'm going to do full cats. I'll probably do. So this is what I was trying to draw on the whiteboard. Obviously a stylized cat. You can draw this if you want. You can draw just the head. If you really want, which would be kind of sad, you could even just draw a round ball and color it in. It doesn't really matter. Right, so this is our black cat. Now, obviously, drawing a black cat on a whiteboard is kind of tricky because if you draw it all in black, well, then you can't see any of the features. So I'll just color in the paws. You know, when I was growing up, there's a TV series by a guy named Hart, H-A-R-T. Should have to dig it out sometime. And he used to do drawings like this. So it's kind of funny that I'm now emulating them. This was from like the 80s, right? Before many of you even were around. So anyway, black cat in sort of 3 dimensions. Going to be crossed. Oh, and also this has, let's use the letter B, not beta, because you have to be super careful with Mendelian genetics. This is why I don't like using letters so much. I prefer the crosses and stuff, because obviously upper and lower cases have to be different. And then over here we have, this one's going to be a little scratchy because I'm going to be stretched out now. Mm. As I like to say, any differences in what the cat actually looks like other than their color. So, you know, some of them have bigger butts basically, or fatter tails and stuff like that. Right. It's purely because of my failings as an artist. It's not because there's any difference in other phenotypes. Just wanted to add that, you know, caveat there. So this individual is going to be, oh, actually, question for the class. What's the genotype of the purple cat? Using the same letter format that we've used for the black cat. Correct, everybody. It is homozygous, recessive. Right, because the parents are always true
Professor,
breeding. Hello?
is the cat black and purple or just purple?
No, it is. It
Oh,
is
sorry.
actually-
It looked black and purple from where I
No.
am.
That's all right. If it was black and purple, then we'd be looking at incomplete dominance or actually co-dominance, which would be something completely different. So this is really as simple as it kind of gets in terms of phenotype. So if there are any kind of weird ass differences because of the light or my drawing or whatever, it really is as simple as black and purple. All right, so these most beautiful cats, are going to produce gametes, right? And I like writing out gametes because it kind of like tortoise versus hare kind of deal, turtle versus hare, never sure which. Do this slow, it'll always work, right? So this parent can only ever produce gametes with a single dominant allele, right? Because remember, the adults, the individuals are diploid, their gametes are haploid, always and forever, right? One hopes. Same applies here. Homozygous recessive, so the only allele that this individual can produce in its gametes is a lowercase b. All right. So my apologies for having to go through these multiple panels to do a single cross. It's just the challenge of this format, I guess. All right, so everybody got that okay? You're caught up on your drawing? Yes, Joshua, that is 100% correct. So the only individuals that these two animals can produce are going to be heterozygous offspring, and they're going to have same phenotype as the dominant homozygous dominant parent. That's exactly right. That's why I like splitting it out into the alleles, because essentially when you're reproducing, right, you're adding one allele of yours to one allele of your partner's for any given gene. So you can only ever get, you know, that outcome. And that's right, Trinity. So the offspring, because they're heterozygous, will carry the recessive allele, but they won't express it. Right? So they will have it there, but you won't see it because it's going to be masked by the dominant allele that it got from its homozygous dominant parent. That's also another trait of recessive, or normal recessive traits, is that they skip generations, right? So they disappear for a while and then reappear. So in the next generation from these two, this phenotype will disappear, right? Because you'll only ever see the dominant black cats, but it will reappear in the following generation. That's just another way of thinking about it. Did I take a picture of that? I think I did. I did. Okie dokie. So if we kind of imagine that we're scrolling down, we have our gametes from our parents. Those come together to make our dominant black cat in the F1. Like so. Right? That's the only genotype you can get, and phenotype. So these are going to be black heterozygous cats. And that's the F1 generation. Now, I kind of didn't leave myself enough space down here, which is a bit of a pain. But question to the class: what possible gametes can this individual create or contribute to the next generation? Right, it's going to be heterozygous, but only one of these alleles goes into each gamete. Right, exactly. So it's either going to be a dominant B or a recessive B. Now, let's go back to the assumptions that we talked about last class. What ratio would you expect to find these alleles in? So if you were to take, I don't know, 100 gametes, let's say, from this individual, whether it's a male or a female, and you look at which allele they have, how many of those 100 would you expect to be a dominant allele?
50.
50, right. And the same for the recessive allele, because that is Mendel's law of segregation. Right. Good job, Samantha, you too. I didn't actually catch who that was that spoke, but good for you as well. So law of segregation states that which gamete gets which allele is totally random. So if you have two alleles, it's essentially a 1-to-1 or 50-50 chance that you get one versus the other, right? So actually, you know what? I'm gonna truncate this cat. There you go. Again, it's a short cat, but the only phenotype we care about is the fact that it's black. So this is a heterozygote, and it's going to generate big B or little b gametes. And that's going to be in a 1-to-1 ratio, right? Or 50-50 big B to little b. And so that's a key assumption about this cross, right? It has to happen this way, or it's not a Mendelian cross. Now things get a little bit more complicated. So before I go any further, and I'm not picking on you, Haley, I just want to check. Hopefully, Haley, this helps with your previous question about what is going on when we look at it in more detail like this. Right. So again, this is like another way, right, of looking at the same stuff. So these individuals are going to be reproducing with each other, right? Which in terms of genetic diversity is a terrible idea and you shouldn't do it. But in terms of Mendelian inheritance, this is it. Hey, no worries, Haley. That's the whole process. That's what we're here for. Okay. So essentially we have two individuals. F1. F1, right? Shorthand cats. And these can produce either big B or little b gametes, right? Because they're heterozygous. It's funny, every time I say Big B, I think of Train Spotting. It was a name of a character from that film. Okay, so in terms of figuring out the F2 generation, right, which is what all of these are going to be, all you do is you simply add these together. That's it, right? That's essentially what reproduction is. We're kind of boiling it down to the basics. So you have two B's, you've got a big B and a little b, and we always write the dominant allele first just from habit. I don't know why, but we do. And then the final one will be homozygous recessive. So the genotype ratio is going to be 1 to 2 to 1. But you can't see the genotype ratio. All you can see are the phenotypes, right? And so this individual is going to be black because it's homozygous dominant. This individual, if we had 4 cats, right? It's also going to be black because it's heterozygous and it has 1 dominant allele. We're going to have the same over here because obviously it's the same genotype. And then the only one that's different is here, with my unfortunately very similar color, purple. So you can kind of see a little bit better when it's zoomed in. Right? And that's what gets us our 1, 2, 3. To 1 ratio. Right? And that is what you'll always get from a monohybrid cross. All right. Have a quick cogitate on that and ask any questions you want. And it does sound kind of cheesy, I know, but I am very happy when students tell me that they don't understand what I'm talking about, because it gives me a really good opportunity to think how I can improve that and help you get it, because that is really what gets me out of bed in the morning. Well, and the fact I've got to feed my dogs as well. That also gets me out of bed. All right. Did I take a picture of that? I don't think I did. Let me check. Oh, I did. Any questions? Okay. So a dihybrid cross, which we're going to start, but we're definitely not going to finish it today. We'll finish it on Thursday. A dihybrid cross is exactly the same, with the exception that we now have 2 genes and 2 combinations of phenotypes in the parents, and the F2 generation is way more complicated because you have more things involved, essentially. Everything else is the same. So the big difference between a monohybrid and dihybrid is this bit, the F2 generation. I was quite pleased how that came out. All right, are we all good to move on? Speak up if not. All good. I even got a thumbs up there. Awesome, Matthew. That's fancy. Okay. All right, now I am going to inflict on you the long-haired cats, which are my personal favorites, I have to say. So, dihybrid. We now have 2 genes. Each with 2 alleles and really phenotypes. Right, so each trait or gene is going to have a dominant and recessive allele, a dominant and recessive phenotype, right? So potentially in the F4 generation, the F4, the F2, we're going to have 4 different combinations of phenotypes. Right, that's where the 9 to 3 to 3 to 1 ratio comes from, because we've got 4 different ways in which we can mix up these potentially 4 different phenotypes, right? 2 for each gene. So now, now let me get this straight. Oh man, I should have double checked on this beforehand. Give me just a second. I'm pretty sure, because you know what I'm like. I do like to do this correctly. TTh Genetics, Wikipedia. Hair length. Pretty sure hair length is yeah, there you go. I thought short hair was dominant, and I was correct. I just wanted to check. All right, back in action. So I don't want to teach you the wrong thing about cat genetics. That would be awful. So we have, same as before, this is where it gets funky. This one might have had mumps at an early age and been doing lots of squats at the gym. So essentially, I'm drawing out exactly the same thing that I was earlier. Right, very deliberately drawn tail there. So this individual is black, obviously, right? And it is a P0, so it's a parent. I've left myself enough space over there. And so its genotype is, as before, big B, homozygous dominant. However, it also has short fur. See? Short fur. And so, which all my cats are, thankfully, even though one of them sheds like it's going out of fashion. I'm actually amazed there's any of that cat left now that fur she sheds. And so short fur is dominant over long fur. It's actually down to growth signaling and stopping fur growth, actually. And so, I'm going to use letter L because both S and F aren't much fun when it comes to using Mendelian genetics. Okay, so that's the dominant. All right, so what are the phenotypes of the other parent going to be? Question to the class. Got fur color and fur length. Yeah, there we go, Samantha. So that's the genotype, Samantha. Let's write that down. And then we can work from that. Actually, Samantha, that is the proper way of doing it. So go from genotype to phenotype. So what's the fur color for homozygous recessive? Based on the previous one, the monohybrid that we did. Purple. Thank you very much. And what's the opposite of short? Long. Some of you may have heard this story before, but actually had a friend who had a couple of long fur cats that she called Watson and Crick. She actually named Crick, the black cat was called Crick, which is really funny if you know anything about the kind of stuff that that guy said. But they shed fur like you wouldn't believe. It was like entering a, I don't know, a pillow factory, just like fluff everywhere. So this cat is going to be purple. And again, size difference is not an issue here. Aha, now it's going to have long fur. There you go. That's what my version of a long-furred cat looks like. You can butcher that picture as much as you want. I don't mind. It's a lot of fun. Okay, so those are going to be the parents in this cross. Now, hope everybody appreciates my long-furred cat. First time I've ever done one in purple, though. Don't know what that is. Now what we've got to do is work out the gametes. So, questions to the class. What gametes can this individual produce? The only gametes that this individual can produce. Remember, you only get one allele for each gene. Oh, that's you. All right. So, and here's the key. It's not two different gametes. It's one gamete with two letters, right? This is, pull it up a little bit closer. Really, really important. Because you cannot lose letters during your cross, right? So you get one of each of those in the same gamete, right? Thank you, Trinity and Samantha. And you get the same deal with the purple long-haired cat. So this individual can only ever produce gametes with one recessive allele of each gene. This is, in my experience, teaching Mendelian genetics for many, 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 many years. This is where students often come unstuck because they forget that the gametes are haploid. So you can only have one copy of each gene in the gamete. Quite often, students will put two different alleles, one which is double B, the other one which is double L. And so essentially you're losing a gene in the process, right? Your gametes have to have a copy of each gene in the cross, but only one of each, right? That's super, super, super important. Otherwise, the whole thing just falls to pieces, doesn't work. All right, so what phenotype combinations is the F1 heterozygote going to express based on this? Trinity, that's correct, but that's the genotype. What's the phenotype? Or phenotypes, rather. So you're 100% correct, but that's not what we see. There we go. It's going to be black and short fur. Technically, it's fur, not hair, if I'm going to be super, super picky, Trinity, but it doesn't actually matter. All right, so did everybody get that? F1s are going to look like the dominant parents. So these, we're going to do the same as we did for the monohybrid cross. We have almost did it myself. There you go. Right. These come together. That's going to give us use my cat drawing mojo. This is a little bit like is it Nermal? Garfield's cousin or something like that. I can't remember. I'm not an expert on Garfield, even though I do love cats. I might need to kind of gen up on that a little bit. Right, so this individual is going to have, as we have seen before, a big B and a little b, and a big L and a little l. Right, this is the F1 generation. All right, so it expresses the phenotypes. So this is going to be black. I'm leaving space down here because I need it for the alleles. Black and short fur. Right, those are the two phenotypes that this individual has. Okay, so now the question is, and this is where things start getting complicated. What gametes can this individual produce? I'm going to give you a hint. There are 4 different gametes. So you can start anywhere. Just pick 1 letter from each of these and just start. Someone give me the first gamete. Oh, thank you, Samantha. So here's the first one. And I'm glad you put up that Trinity, not because I want to poke fun at you, because I don't at all. But that's a very, very common mistake students make when they first do this, right? Is rather than splitting these, they kind of group the alleles together by gene. That's not how it works, because each gamete can only have 1 allele. So each of these, 1 allele is going to go into 1 gamete, the other one is going to go into a different gamete, because the gametes are haploid. They only have 1 version of each gene, 1 copy. So as Marissa said, next ones are big B, little l, right? And Matthew has little b, big L. And then we have the small ones of both. Little b, little l. And so essentially it's all possible combinations of these. I'm going to bring this up a little closer because it is super, super important. So we have big B and big L, right? These two. give us this one. Big B and little l over here give us this one. Then we do the same for the recessive allele for fur color. Small b, big L, give us this one. Small b, little l, give us this one. And that's how we get what Matthew wrote, which is the 4 different possible gametes, each with a different combination of dominant or combination of the 2 alleles. So dominant, 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 recessive, recessive, dominant, and recessive, recessive. All right. This is where students come unstuck with dihybrid crosses, right? It's super normal. Don't worry about it if that happens to you. But let's just make sure we get that corrected, right? Essentially, or fixed before the exam. That's the key. Doesn't matter if we get it wrong before then, as long as we get it fixed by then. So yeah, those are, I'm not going to, so I've got a question that those are the 4 genotypes. I'm not going to call these genotypes because they're, I'd like to use the term genotype for what you get in an individual rather than what you get in a gamete. So what I'd call these are 4 different allele combinations, right? 4 different ways in which you can mix up these 2 genes, right? So yeah, but there are definitely 4 different combinations that you can get. All right, so good, glad that helped. What was I going to say? All right, so law of segregation is what? What does the law of segregation tell us about where the alleles go? Come on, gotta be off the tip of your tongue. What does the law of segregation tell us about partitioning or segregation of alleles? Yeah, so which of these alleles for this gene go into which gamete is random, right? So essentially 50% of these gametes is going to have a dominant B, 50% will have a recessive B, and that's what we get. We get 2 dominant, 2 recessive. All right, so that's the law of segregation. Now the law of independent assortment. Oh, there's my second child. She is dressed for the weather. She's got like on, you know, thermal gear and stuff. It's she takes after the mother. Anyway, but now we have 2 genes, right? And so the law of independent assortment, in addition to the law of segregation, says that the allele you get or the genotype you get for this gene doesn't affect this one, right? And so those 2 together again mean exactly the same as for monohybrid cross. Actually, I wish I had a different color. Green work. Maybe. We have a 1. Let's put it here. 1 to 1 to 1 to 1 ratio. So we get those 4 different combinations in the same proportion, right? So essentially 25% of them will be this one. Yeah, this is the law of independent assortment. So the law of independent assortment also applies later, but it is worth talking about here too. Essentially, it says that these genes are independent, right? So the gene for black fur, well, gene for fur color and the gene for fur length don't interact, right? They are independent from each other. They don't affect each other. And so that gives us this 1 to 1 to 1 ratio of gametes. Okay. Now what I'm going to do is I'm going to set up the next step, and I'm going to finish there, because that will be A, time, and B, I need to make myself a coffee before we start that. Okay. Kind of the hope in a sense is that I get back to the classroom before I have to actually work this out, because it's going to be hard work on such a small board. So we have 2 individuals, right? Again, we have our F1, black, oops, a bit too big, black and short fur, here and here. Each one of those produces the same potential combination of gametes. So we have this, 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 and this. Right? And the same for this individual. Now, when you, and this is going to generate, I don't know where I'm going to put it. Let's put it here. Aha. That's going to generate the F2s. And what you need to do, and I'd suggest you do it by next class, it won't take you very long. Add these together. So I'll give you the first one. This one is going to be homozygous dominant for both. And so it's going to be black and short fur. What I want you to do is to add these together. So it'd be, let's give you another one. Oops. Big B, homozygous dominant, big L, little l, heterozygous for fur length. But because we have the dominant fur length allele, it'll be a black short fur cat. So finish this off yourselves, and we'll go over it again on Thursday. All right, I think that's probably enough for now. Anybody that's in my lab section next, I will see you back in Zoom in about however long it takes me to make a cup of coffee, which will be about 15 minutes. And we'll go over lab. Any of you that have Miss Vaughan's lab this afternoon, she should be sending around the same kind of thing that I did with a Zoom link of her own. It's going to be different from this Zoom link because obviously she has her own Zoom account. So she'll send you her Zoom link to meet. And as before, we'll play it by ear for tomorrow. All right. See some of you in a bit and the rest of you on Thursday. Take care, everybody. Ooh. Hello, love. Here's a very long shot.